Thank you very much for joining us today. For those of you that don't know me, and for some, thank you online as well for joining us. So my name's Felicity Gavins. I'm the director of the CIRTM, the Centre for Inflammation Research and Translational Medicine, and I'm delighted that today we're able to showcase some of our research for Brunel's Research Festival. The Research Festival itself is going on for a month, so please immerse yourselves in research that's going on around Brunel. So this afternoon we've got a very nice and diverse lineup of talks from members of CIRTM, and I'm delighted to welcome the first talk from Doreen Lau, who's going to talk about the role of low avidity tumour-specific CD8 T cells in cancer immunotherapy. Thanks, Doreen.
Thank you, Felicity, for the kind introduction. Good afternoon, everyone. I'm a new lecturer in Biosciences Division. I'm also an immunologist known for using bespoke imaging approaches to understand how our immune system fight against cancer and using chemical and engineering approaches to improve the delivery of drugs into the tumor microenvironment so as to improve immunotherapy response. So the work I'm presenting to you today is something I was previously involved in Oxford on investigating the role of low avidity tumor-specific T cells in cancer immunotherapy. So immunotherapy, as we know, has revolutionized the way we treat cancer. Various immunotherapeutics such as cancer vaccines, CAR T cell therapy, as well as immune checkpoint inhibitors has been developed over years to treat patients with cancer. Immune checkpoint inhibitors such as CTLA4 and PD-1 have increasingly been used in clinics to treat advanced cancers such as malignant skin cancer, metastatic melanoma. Although it's increasingly being adopted into clinics to treat patients, not all patients actually benefit from treatments because of underlying differences in terms of the tumor immunobiology. And even some patients may actually develop a relapse or treatment resistant on therapy if although T cells may be present within the tumor microenvironment. And why is this so? So the way T cell recognize these cancer is via specific interactions with between the T cell receptor and the tumor antigen presented on class 1 MHC molecules on cancer cells to these T cell receptor. So this is called antigen presentations. Antigen presentations is a process that requires a lot of fine-tuning, not just based on whether the antigen is the right match for this T cell receptor, but also how strong the binding strength is in terms of these interactions. Because if the binding is too strong or the signaling is to a certain extent where it causes T cell chronic stimulation and exhaustion, the T cell may not be able to function in the longer process during this long marathon fight of attack against the cancer. Furthermore, T cells may experience persistent antigen exposure within the tumor microenvironments as the tumor develops. There's a greater antigen load, and this may lead to chronic T cells stimulation and exhaustion. So T cells recognize cancer based on the affinity match between the T cell receptor and the peptide sequence on the antigens. However, each T cell can express more than one T cell receptor for interacting with multiple peptide MHC molecules on the cancer cell surface. And together with the expressions of co-stimulatory and co-inhibitory molecules, as well as adhesion proteins on the cell surface, this contributes to the overall strength in terms of binding between the T cells and the cancer cells. And this term is called T cell avidity. So T cell avidity actually can contribute to a certain extent the quality of the T cell receptor signaling, the activation of T cells, and this process of whether it's able to sustain its approach in killing the cancer cells by releasing cytotoxic granules to kill the cancer. So to be able to study this process in further details within the living system, we need a good model. So we use a CT26, a preclinical model that called CD26 mouse colorectal cancer that express a model antigen that called GSW11 that's derived from glycoprotein that's expressed in more than 20 types of mouse cancer cell lines. And this leads to the priming effect of to have a natural occurring endogenous T cells, tumor-specific T cells, where we can track the identity with specific biochemical stainings and also track these antigen load over time within the tumor microenvironment as the tumor develops. And also to understand changes in these T cell ability in the tumor-specific T cells as the mice are treated with immunotherapy using PD-1 inhibitors. So to study T cell ability especially is an approach whereby you want to be able to understand the force measurements in living cells. We partner out with a company, a spin-out company from VU Amsterdam called Lumix to perform direct biophysical measurements of T cells interactions with cancer cells, which presents an antigen. So this company, Lumix, actually have engineered very interesting instruments, which consists of a microfluidic lab-on-chip device that allows you to perform co-culture the T cells and cancer cells together, allow them to interact and on the living chip for several hours during your assays. And the instruments also consist of an ultrasonic device, a transducer, which can ramp up increasing force of acoustic force on the chip to pull these T cells and cancer cells apart from each other. So it gives you a relative measurement of the binding strength between the T cells and cancer cells on the chip itself. You get this single T cells measurement from each of these T cells you try to incubate together with the cancer cells. A very interesting aspect of this instrument is it's also a microscope. So it has a bright field interface where you can visualize T cells interactions with cancer cells. For example, here, searching for the target, the cancer cell, you can see all this Filippo kind of things trying to save up the cancer cells and also the cardiac contact together. To be able to track this T cells, its movement on the chip, the T cells were labeled with a dye that can be detected on the fluorescent microscope in the far right channel using the microscope. And based on semi-automated image analysis of this T cell bouncing videos, you can derive single T cell detachment force, the force at which each of these T cells actually bounce off from the cancer cells and generate a curve called the T cell ability curve for each of these different groups of T cells. So as shown over here on your far right, when we try to do a comparison between T cells isolated from tumors that respond to treatments versus those that didn't respond to treatment, there's actually a greater population of T cells that are of lower avidity in tumors that responds to treatment with PD-1. So to confirm this, we perform traditional immunostainings with peptide-MHC tetramers, which allows us also to perform some kind of assays to see the T cell avidity. So what is confirmed is it shows that within the tumors that do not respond to treatment, it only contains low avidity tumor-specific T cells, as you see over here in this cartoon. And in tumors that do not respond to treatment, it either contains high avidity T cells that's more terminally exhausted or no tumor-specific T cells at all. So it's not very immunogenic in a sense. We've performed further functional analysis of these different high and low avidity T cells populations, which showed that the low avidity T cells are actually have exhibit a more precursor exhausted or less exhausted phenotype compared to the high avidity T cells, which are more terminally exhausted. The low avidity T cells have a lower co-expression of CD39, PD1, and EOMs, and it has some kind of progenitor stem-like features with a high expression of Tbet and TCF1. To be able to examine the cancer-killing cytotoxic ability of these low avidity T cells versus high avidity T cells, we perform cytotoxicity assays both on in cell culture as well as within the in vivo mouse model. So using a T cell and cancer cell co-culture system, we showed that these low avidity tumor-specific T cells are actually more professional killers. They are able to do a cleaner job at killing most of the cancer cells and driving most of them towards the late apoptosis phase as compared to the high avidity cells, which only manage to complete the job less than halfway through before they become terminally exhausted. When we inject these low avidity cells within the in vivo system into mice, together in combination with PD-1 immunotherapy, tumor regressions and or tumor melting away of complete cure could be seen in this mice. So it does bring some kind of promising approach using low avidity cells to cure cancer. Further analysis of the bulk RNA-seq data showed that the low avidity cells have a significant upregulation of 1054 genes and downregulation of 17 genes as compared to the high avidity T cells isolated from the non-responding tumors. Gene set enrichment analysis of this data showed that these tumors, the low avidity tumor-specific T cells are significantly enriched for pathway associated with T cell signaling, cancer-killing cytotoxic ability, and also metabolic energetic functions like oxidative phosphorylations. Some of these genes include granzyme B, FASL ligand related to its ability to kill the cancer, and there's also ICAM1 related to adhesion between cells, important during this target contact process, and also some other genes related to antigen presentation and T cell receptor signaling. A very interesting aspect is some of these gene signatures map to cat disease pathway associated with allograft rejection, autoimmune disorders, and viral-mediated diseases. So in conclusion, T cell avidity may influence T cells' ability to recognize cancer and sustain its attack on cancer cells. In our study, we found that low avidity tumor-specific T cells are preferentially expanded in the responders to PD-1 immunotherapy. These low avidity T cells are more professional killer than the high avidity T cells, and this may be attributed by the stem-like quality. They're more energetic when they and able to sustain the activity in killing cancer. So future study may consider, considering a vaccination approach or adoptive transfer of the low avidity T cells to improve immunotherapeutic approach. So I would like to thank my teammate for for the effort in contributing the studies from Oxford and Southampton, as well as Lumix for designing a very interesting instruments. And special thanks to my funders, Worldwide Cancer Research Grant, CRUK, as well as Wilson College for supporting my fellowship in Oxford. And in my new group here in Brunel, I'll be continue to focus on the theme of immuno-oncology and taking a step further to understand cancer immunity, the mechanisms underlying it, and response to immunotherapy. And furthermore, tapping on my background in imaging science, chemistry, as well as engineering, I have been working with collaborators to find better ways to design better probes to monitor how T cells infiltrates into tumors on the whole body scale, to examine what's happening between T cells to cancer cells, as well as other microenvironmental factors influence interacting with each other on the microscopic level, and also certain ways to package drugs so that we can improve their delivery into tumors to improve immunotherapeutic outcomes. So if you are interested to in collaboration or PhD in a related area as this, please feel free to contact me or follow me on Twitter. With that, I thank you very much for attention.
Doreen, a lovely talk. Very interesting. We've got time for a quick question if anybody wants. Sorry, Christina. First time.
The long-lived cells, what's the role that they normally play in a response against pathogen? Are they also better, or is it something that is particularly useful in cancer because somehow the cancer has less ways to infect?
I think in terms of chronic inflammation, chronic infectious disease, as well as in the context of cancer, where the antigen load is quite high and persistently there, having the ability of the T cells having the ability to detect, to interact and slowly launch this attack on cancer is very useful. But if it's too many antigens and it has a strong binding to this antigen, it may lead to its ultimate exhaustion over time because it's an auto feedback mechanism.
So they're really useful in
Chronic disease context. Yeah, that seems like HIV also is the same case.
Thank you. We don't really have time for any more questions, sorry. But after the talks, we're going to do the poster judging. So hopefully more discussion then. Thank you, Doreen. Okay, moving on to our second talk, which is going to be given by Sophie, my PhD student. So she's going to talk about the therapeutic potential of Annexin A1 derivatives to promote inflammation resolution. Thank you, Sophie.
All right, so today I'm going to be talking to you about inflammation resolution and a potential new therapeutic avenue that we can use to treat inflammation. So, inflammation is your body's mechanism to heal damage. But what happens when you have too much of a good thing? Chronic inflammation is present in multiple different diseases, such as cardiovascular disease and stroke. Despite the therapeutics available on the market currently, they are the first and second leading cause of death globally, and they account for around 15 million deaths annually. So when you think about treating chronic inflammation, anti-inflammatories might come to mind. So this is preventing the initiation of inflammation. But now we know that inflammation is actively resolved, which means that we have a new therapeutic avenue to look into. Inflammation resolution means that you don't suppress the initiation of inflammation, and therefore you wouldn't be suppressing the immune system of the patients. So that would mean that if you're treating resolution inflammation, you're trying to promote the resolution of inflammation, then you basically don't have to immunosuppress these patients. You can keep the good inflammation without having the bad chronic inflammation, basically. So one of these mediators for the inflammation resolution of inflammation is Annexin A1 and its corresponding receptor FPR2. So Annexin A1 that you can see on the right-hand side has a core domain, and then the yellow protein structure is the N-terminal domain. The N-terminal domain is responsible for the majority of the signaling, which actually means that we can take N-terminal derivatives of this and retain a lot of the signaling properties of the entire protein. Now we open ourselves up to how long of a peptide derivative do we need. So the different lengths of the peptide derivatives can have different changes in the receptor signaling. So you can see there we've got an AC2 to 26 and we've got an AC2 to 50. So there's a lot of different peptide lengths that you can try, but so 2 to 26 has been looked at in the literature a lot. 2 to 50 is relatively new, and we are looking at how we can characterize it. Because as I just said, 2 to 26 won't necessarily have exactly the same receptor signaling changes, for instance, as 2 to 50 will. So the receptor FPR2 is actually expressed very highly in cells called neutrophils, which are your specialized cells of your innate immune system. These cells specifically have functions such as mitosis, leukocyte transmigration, reactive oxygen species production, and myeloperoxidase release, which is an enzyme. So these would be like your first responders, essentially. If there is a signal for damage, they're usually on site first. Because they have such a high expression of FPR2, it means they're actually a very good target for the AC2 to 26 and AC2 to 50, or any of the other Annexin A1 peptide derivatives. But as I said earlier, we still don't necessarily know the reaction that the neutrophils will have to the AC2-50 peptide. So we need to characterize treatment with the AC2-50 peptide under all of these functions. So how I did this was by taking neutrophils from healthy donors, using a histopache 1077 and 1119 density gradient, which essentially means because your neutrophils are heavier, they're more likely to go into the 1119 section. And that means you can get a relatively pure population of purely neutrophils to then test by pre-treating with the drug, stimulating them so that they're in an inflammatory condition, and then you can test how these neutrophil functions are changed. So first of all, I can go into the transmigration function that I just mentioned, which is where if you've got a damaged site, then your neutrophils will recognize signals in that damaged site and then move towards that site, which is what chemotaxis is. So with the AC2-26, it's already relatively known that at certain higher concentrations, it does reduce chemotaxis. But as you can see with the AC2-50, we now have a set of in vitro concentrations that the chemotaxis is reduced by, which are actually relatively similar to the 2-26. So there wasn't too much difference there. The next function that I looked into is the netosis function. So netosis is a neutrophil extracellular trap formation, which is basically where DNA is either released in vesicles or it's released extracellularly by two different pathways. So you can see that there is the non-lytic, which is where the cell membrane, the plasma membrane is retained, and DNA and mitochondrial DNA can be released. Whereas you have the lytic, which is with a different stimulant, and that's where the entire plasma membrane basically disintegrates. And you'll see a gradual increase of netosis, whereas this one is basically in installments. So very different pathways, which is why we need the two different stimulants to study them. Here you can see that within this pathway, generally mitochondrial ROS is stimulated with the non-lytic netosis, whereas with the lytic netosis, for instance, it's more total ROS. So we'll look into these different aspects. What I found is that AC2-26 does not significantly alter netosis, neither with the non-lytic nor with the lytic. And that was across the entire time series that I did with both of the stimulants. Whereas with the AC2-50, it is significantly reduced. And because obviously we don't know the concentrations, I did a concentration titration to get the appropriate, which was 100 nanomolar. So we can see that it works with the inomycin, which is the non-lytic, but clearly it doesn't work with the lytic. So now we're starting to distinguish exactly what kind of neutrophil functions that this peptide is actually beneficial to, and also what pathways it clearly is impacting. So as you saw in the pathways before, I've mentioned reactive oxygen species production, which is important for both of the stimulants. With AC2-26, the inomycin stimulation, considering we know that the AC2-50 did significantly reduce this, funnily enough, the concentration is exactly the same as previously, and it's showing a decrease in reactive oxygen species, whereas 2-26, despite not actually reducing netosis, is showing a significant reduction in reactive oxygen species production. There is some literature depending on how important the reactive oxygen species production is to the netosis as well. So this could be a factor to include. So ultimately, what I've established is a concentration that is efficacious within these in vitro assays, found exactly what functions are actually significantly reduced within the neutrophils, and how these concentrations differ between the functions. So as you can see with chemotaxis, much higher concentrations are needed to be able to significantly reduce it, whereas with netosis, a much lower concentration is needed. Netosis in itself is also only significantly reduced with one of the pathways and not the other one. So this kind of opens up a lot of different avenues to look into more and further explore. All right, thank you.
is there any disease context where you're investigating these effects of different peptides, or is it just a basic sort of?
So we're starting in general as an inflammatory context. We are then moving into disease context. It's just that we don't actually, there's not really much around. It's better to start with an efficacious dose in mind before you move into those disease contexts. And also it gives a good idea of exactly, because when you start going to, for instance, we're looking into stroke and another group is looking into cardiovascular disease, it's a good benchline to start with to then consider what other diseases it could be involved in as well, because Annexin A1 isn't just within those two that I mentioned, it's within cancer and all sorts. So it's just we didn't really have the basic information there to begin with, so we have to generate the experiments.
Intention to go into studying cancer or cardiovascular disease or any.
Oh no, we are going into stroke.
So you're
Yeah.
doing this.
Yeah, into ischemic stroke. Yeah. So that will be both in vitro and in vivo mouse models. Yeah.
Obviously, looking at stroke, is the plan to do prophylactic treatment and post-stroke treatment, or have you not thought about that yet?
So with the nature of stroke, because the majority of people will be, like patients, for instance, will be rushed to hospital and then they'll have either a drug called TPA administered to be able to dissolve the clot, or they'll have it surgically removed, or other options as well. But so prophylactically is potentially an option, but not what we're going to go into. We're going to look at the point of clot removal, reperfusion of that area, which would then be when the drug would be administered.
Okay, thank you very much, Sophie. Yes, fine. So I shall hand you over to my co-chair, Gavin. That's on the phone.
So thank you, Felicity, and I'm delighted to introduce the next speaker, who is Ekuwa Sakyi Adade, who's been looking at examining the impact of climate variability on malaria case rate in Ghana.
Thank you very much. So my name is Ekuwa Sakyi Adade, and my entire PhD is focused on examining a climate-sensitive disease, and my focus is malaria. Malaria is one of the top 10 killer diseases in Africa, in Ghana as well. And my focus is Ghana, where I come from, and mostly pregnant women and children under 5 risk being killed by malaria. And so though it's not too common in the West, it's very, very common in the African continent. This is just a part of what I've done so far. So it's just a section of the data analysis that I've done. Yeah. So the theory I'm building up on is on the fact that climate and weather variables directly affects the rate of malaria transmission. And then the effect is rather reduced by the influence of both healthcare responses and socioeconomic structures. And so in effect, if there wasn't the influx of healthcare and access to pharmacy and things like that, we would rather see a lot more cases of malaria. Since the climatic conditions at different geographic areas are varying, it's important to understand how much of an impact climate change or the changing climate would have on malaria at different geographic different places, different geographic areas. So a bit of malaria in Ghana, the period of transmission is found to be dependent on the dry and wet season. Unlike here that we have 4 seasons, maybe, in Ghana, we have just the dry and the wet season. That's what is recorded. And so the period of transmission of malaria is very dependent on these seasons, and high rate of malaria cases in the northern part of the country is recorded between July and November. Again, in Ghana, we have the northern middle belt and then the southern belt. And so in the northern belt, we have high rates recorded between July and November. And in the southern belt or middle belt, we have seasonal transmission between May to November, and less cases recorded from May to June, high cases recorded from October to November. My data was from the Ghana Health Service, the malaria data. It covers 261, all the 261 districts in Ghana. We have 16 administrative regions, and these are further grouped into 261 districts. And my climate data is also from the Ghana Meteorological Agency. They pick the signals from 22 synoptic stations. So first, we look at the national case rates. Those are the incidence rates, and then also the absolute cases. We noticed a sharp rise from 2008 to 2013 in the case rate of malaria, and then we have the sharp decline and then the gradual fall up till this time. It's important to note that around the same period, there's been intervention, strategic intervention policies going on. We have treated mosquito bed nets being shed. We have the RDTs. If you start feeling feverish, you go to the pharmacy, you report, you check if you have malaria, and then they start treatment. And so very much aware of the socioeconomic factors that are at play, what is going on to prevent malaria. But this particular work focused on what climate could be doing to the disease change. And so this is the pattern we noticed. And we also investigated the data we have at the administrative regions, and we see quite same pattern where we have the rise and then the sharp decline. And so malaria has steadily declined since 2013, which is very commendable. Since we are looking at the influence of climate, we specifically looked at zoning that had been done, and we picked Bisa et al., who used cluster analysis to do the zoning of the climatic conditions in Ghana. So he divided the country into 3 zones. We have the savanna, the forest, and the coastal. And so it's pretty much like the first scenario that I gave, that the country is divided into 3, the northern belt, the middle belt, and then the southern. So that's the same thing that he's done, but he uses the 22 synoptic stations. And so it was very relevant to the study. And so we moved on to look at the malaria case rate according to these zones. And what we did was that the 261 districts, we picked the geographic locations that falls under these zones, and then we put the totals together, and then found out what the malaria case rates looked at around these zones. And we see the same pattern also repeat itself across all the zones. This is the temperature variability. We've done the temperature, rainfall, and humidity, but for the purpose of this presentation, I'll be looking at temperature, since temperature is rather, let me say, though negatively correlated, it was stronger in the correlation analysis. So I want to focus on temperature variability here. Temperatures above 34 degrees have detrimental effects on the survival rate of malaria parasites. And so we looked at all these 3 zones. The pink represents savanna zone, the green, the forest, and then the blue represents the coastal zones. And we realized that the savanna zone records temperatures at a certain time above the optimum range of temperature, though the forest and savanna is within the optimum ranges. So we picked out that indication that the savanna zone has some seasonalities where malaria transmission may be very, very low due to the fact that the climate condition around that time is not favorable for the malaria parasite. And so we moved on to look at the annual cycle. We looked at January to December, and we took particular attention to the period June to October, where earlier on I stated that this season records the most high cases of malaria. And so when you look at, we grouped it into the two. We grouped it into the two periods that the patterns are very visible. And so we looked at 2008 to 2013, and we look at 2014 to 2022. Yeah. And these are this records the variability, the difference, where within 2008 to 2013, we see by 2014 to 2022, we see surge in the climate variability, in the temperature variability of the savanna area. And so if the average was 35.4 in January, now we are recording 36.2. If the average was 37.2, now we are recording 37.6. So we see close to 0.5 to 1 degrees Celsius increase or decrease between 2008 and then 2014 to 2022. Okay, so we will repeat the same for the other two climatic zones. And the conclusion is that we identified the most important months in the transmission cycle, which is May to June, to October to November across all climate zones. June to October records optimum temperatures across all the three climatic zones, but November to May records temperature out of the optimum range at the savanna climatic zone. Now, when you look at the November to May, between 2008 to 2013 and 2014 to 2022, there's an obvious increase in temperature. And so if ordinarily the increase in temperature between the earlier years affected malaria case rates, then we suggest that it means that the impact on malaria transmission would be exacerbated by 2014 to 2022. And so this is just the first part of the analysis that's done. We are going to use more sophisticated statistical analysis to understand what exactly went on between the period, and then also do modeling studies to see the effect of the climate variability on malaria. Thank you.
Thank you, Ekuwa. So we've got time for a question or two. If I could start, so you showed a nice, dramatic increase in the malaria transmission rate and then a drop from, I think it was 2012 onwards. And you explained why there's been a drop and there's been malaria control measures put in place. Yeah. But what drove the increase?
Yes, so according to literature, you would realize that from that period, 2008, the interventions were now starting, right? So it would be easy to say that because there wasn't that measures put in place, that's why we had the rise in the cases. But this is also something that my supervisor picked up. And now we focused on the decline and realized that the climatic variability was good. That is why we could potentially have. So, but now we are going back to check what really also happened around that time in terms of the climatic conditions that could have probably caused the surge.
Might it be a reporting issue because people started intervening, so then they were measuring malaria more comprehensively than they were from 2008?
It could be, but before 2008, we didn't have the reporting done appropriately like now. So the actual data that I have is from when the reporting itself started. That's from the 2008, yes.
Thank you. Raha?
Thank you very much. My question is about the kind of intervention that you guessed can be done in relation to data and competitive.
Okay, so currently, for example, we have something called the seasonal malaria prophylaxis that's given to children under 5 and pregnant women around the wet season. And now, because of the change in climate, you realize that the period where it used to rain in Ghana is not raining again around that period. And then the dry seasons are becoming wet seasons, sort of. And so this is very important for noting what has changed and can potentially affect the vaccination period that they used to give the prophylaxis. Yeah, so this is one thing that can be done. And another thing is the climate proving the malaria strategies. And so getting to the period where we realize that the climatic conditions are changing, we'll probably start doing the weeding around the giving of insecticide treated nets, like the education and all that, because we are very sure that in the next month or two, malaria is going to increase or decrease.
Thank you very much. I'm not sure we've got time for any more questions, but if you'd like to stay afterwards, there's another question at the back afterwards. Thank you very much. So our next talk is from Kostas Stamatou, who's going to talk to us about suppressing neuroinflammation as a strategy to enhance childhood cancer immunotherapy. Thank you, Kostas.
Can we put like the pointer option on? Hello, my name is Konstantinos Stamatou, and I am going to present you my project with the title Suppressing Neuroinflammation as Strategy to Enhance Childhood Cancer Immunotherapy, which I'm conducting as a research fellow under the supervision of Arturo Sala. Okay. So neuroblastoma is a pediatric cancer originating from sympathoadrenal lineage dysregulation and exhibits high heterogeneity in the disease severity with low and intermediate risk of neuroblastoma patients, achieve 80 to 95% of overall survival rate, but high risk cases only 45%. So approximately half of the diagnosed neuroblastoma cases are classified as high risk, with 20 to 30% of them featuring MECAN amplification. MECAN is a proto-oncogenic transcription factor that regulates many cellular processes like cell proliferation, angiogenesis, metastasis, and more recently suppressing immune activation. In low-risk cases, no treatment or solitary surgery is often curative, but in the intermediate and high-risk cases require treatment. Treatment can be either chemotherapy, radiation, or immunotherapy. We're going to be focusing on immunotherapy. Neuroblastoma has a low human leukocyte antigen HLA type 1 expression, which is limiting the tumor recognition from the T cells. This creates a difficulty in the development of immunotherapeutics, including anti-tumor antibodies, T-cell vaccines, or chimeric antigen receptor or CAR T cells. In the clinics has been used for many years, the anti-GD2 antibody as immunotherapy, as the neuroblastoma are expressing highly the antigen GD2. And this antibody, this treatment is activating the NK cells, natural killer cells, and the myeloid cells in order to improve the survival of the high-risk neuroblastoma patients. However, half of these patients, they don't respond to the therapy. Another therapy that they use as immunotherapy is CD8 targeting CAR T cells. But initially, these CAR T cells fail to enhance the survival, possibly due to exhaustion of the T cells because of the immunosuppressive tumor microenvironment. Recently, they have generated a new generation of CD2 targeting CAR T cells that increased the survival to 60% and also increased the event-free survival to 36%. But this is not enough, and also CAR T cells are quite expensive. So what we are trying to do is to improve the immunotherapy and have a low cost. We're going to be focusing in the myeloid-derived suppressor cells that are present in the tumor microenvironment and possibly they have an immunosuppressive effect. And also in my project, we're going to be using antidepressants or more specifically selective serotonin reuptake inhibitors. Serotonin is a neurotransmitter that regulates many processes, including learning, memory, and happiness. And these inhibitors have been used in the clinics to treat depression by blocking the reuptake of serotonin and increasing serotonin to the brain and helping people with depression. But why these inhibitors? There are evidence that these inhibitors can reduce the MDSCs, the myeloid cells, but also evidence that these drugs have an anti-neuroblastoma activity, reducing the proliferation of the neuroblastoma cells. So the aim of my project is to identify the SSRIs in order to improve the immunotherapy of neuroblastoma patients, validate that these SSRIs have indeed anti-inflammatory activity, and also proceed to a small immunotherapy trial. But which SSRIs? In order to select which SSRIs are going to be using, we conducted an extensive literature research in order to find the SSRIs that they have a possible immune modulatory activity, but also the ones that can be administered to children, because we're talking about pediatric cancer. So we concluded in 5 of them, including one of the most popular one, Prozac. In order to conduct our experiments, this is one of our experimental line. We're going to be isolating bone marrow from mice, treating the bone marrow cells with neuroblastoma supernatant medium, and in presence or absence of these SSRIs, in order to assess whether we're going to have or not reduction of these MDSC cells. But in order to proceed in these experiments, we did some experiments to find the best conditions, and mainly the concentration of the drugs that we're going to be using, because we don't want to interfere with the proliferation of the bone marrow cells. So we did 15 micromolar and 10 and 5 micromolar concentration, and we saw that only 5 doesn't affect the growth of the bone marrow cells. So this is going to be our working concentration. The next thing that we did was to assess whether treating the neuroblastoma, human neuroblastoma cell line with this drug concentration is going to interfere with the growth of the cells. And what we observed was with some of the drugs, we see increase of the G1 cell cycle phase of cells, percentage of cells, which can indicate that the treatment with the drugs decreases the proliferation potency of these cells. The other interesting thing that we observed was that we saw increased cell death in using these drugs. Next, we repeated the same experiment using a mouse cell line, neuroblastoma cell line. But in that case, we didn't observe any cell death, but we did see a strong block and increase in the levels of cells in G1, suggesting that these drugs possibly decreases the proliferation potency of these cells. Next, we wanted to assess whether we're going to be able to induce the MDSCs in vitro. So what we did, it was to have 4 conditions: bone marrow cells with cytokines in order to keep them happy and have the cells growing, bone marrow cells that we just culture them in unconditioned medium, just medium with nothing actually. Bone marrow cells that we use cytokines in order to induce NDSC-like phenotype, and also bone marrow cells that we treated them with neuroblastoma supernatant. From this analysis, we observed that the condition with the cytokines that it can produce MDSC-like phenotype has the same exact phenotype as the condition that we use supernatant from the neuroblastoma. And the other 2 conditions have completely different phenotype and pattern. This is an indication that we can generate MDSCs in vitro. And we can use these MDSCs in order to treat them with our drugs, the SSRIs, and proceed to functional analysis also to understand whether these inhibitors can alter the phenotype of the T cells, but also alter the phenotype and reduce the amount of cells that can be immunosuppressive. And finally, after we have hopefully good results, we're going to proceed the immunotrial that we're going to generate tumors in mice, and we're going to have 4 conditions: control, untreated mice, but with tumors. Then we're going to have mice treated with the anti-CD2 antibody, which is the clinical antibody that is used in order to treat neuroblastoma patients. A condition that is going to contain only the serotonin reuptake inhibitors, and also a combination of these 2 drugs. And hopefully, what we expect it's to the combination treatment to work better than anything else. And this is going to give us the ability in order to suggest to the clinicians to use this combination treatment with very low cost, and help the patients, the neuroblastoma patients, that they are not actually getting help from the current immunotherapy that they're using on them. Thank you very much for your attention.
So any questions for Costa? 10 seconds.
Yes.
Let's talk. Are you planning to test the effect of these plants? Mouse model, and then we each cell. So what you want to focus on your analysis?
So thank you for your question. If your question is if we're going to use human, no, we're not going to use human. We're going to use only mouse-derived, like T cells, and we are planning also to do these assays to see like production of interferon gamma for the T cells. And in the mouse setting, when we're going to conduct the trial, we're going to isolate the tumor and see what is happening with the tumor microenvironment, and see whether we have also like increase or decrease of the MDSCs in the tumor. But mainly mouse.
You don't want to focus on a specific type of cell?
Initially, we're going to do the T cells and try to understand whether we have also production of Tregs. And this is our plan for now. But research can bring other things. Yeah. T cells mainly, because also they use in the clinics, and they're looking to in order to produce like more T cells in combination with the drug, with an anti-CD2 antibody.
OAPC.
Well, the MDSCs, there is a plan to check the macrophages to see how like active macrophages we have or not. But the MDSCs, they contain like neutrophils, like granulocytic type of cells, and also monocytic. So we want to see whether the treatment of this drug, this is the extent like of the project, is to see whether by treating the cells with this drug, we're also changing the monocytic, like the macrophages, if they are active or not. Yes, we're gonna go to that. Yeah. Thank you for your question. Well, the patients that they are not actually respond to the treatment, they are again amplified. But the cell lines that we are using in order to induce the MDSCs, yes, they are again amplified. And also the NXS2 line that we are going to use in order to generate the tumors are again amplified. But these like mouse cells.
Okay, last question, Doreen.
Does serotonin plays any role in this MDSC's activation, migration?
This is what we're trying to figure out. There are some evidence that yes, it reduces. We don't know how. There are some also other cases that they suggest something, but it's not very clear yet. We're gonna try. Like it's like chaotic, field in order to. But we're gonna try to see what is happening there.
Thank you very much, Costa.
Thank you.
Okay, and the last talk for today is from Shuai Zhao, who is going to talk about characterization and biological testing of synthetic electrospun membranes for organ-on-a-chip applications. I'll let you know.
Yeah, hello everyone. I'm Shuai, and I'm one PhD student in CD8. Mainly my project is some of the contents is involved with engineering, but some of them is involved with biomedical. Firstly, I want to introduce what organ on chip. What is the organ on chip? Organ on chip is meaning one application to use for the clinical research, including the drug delivery. But it is also an integrated technology which combined with different technology, including the microfluidics, chips, and electrospinning. About my project is mainly involved with bacterial regenocides. We want to survive with the bacterial regenocides and also some clinical research on this area. And this slide shows how the bacterial regenocides forms. Mainly is involved with the Gardnerella bacteria infect with the cells and another thing I want to mention is that about bacterial regenocides, this disease is very common at the moment and in different countries. It's the intended research about the bacterial regenocides. As you can see, it easily can be seen that the main research is involved with the cells, the cells, vagina cells, but they cannot form with truly and real vagina environment. Another intended research is that some researchers using the animal size as not the real human vagina cells, so they cannot represent the real vagina environment. So the vagina on chip can mainly divided into the 4 parts. Firstly, that to design and build up the vagina on chip platform. Then is the electrospun membrane. It's also, you can call it the scaffold. It's using for the cells to grow and to support the mechanical environment and the mechanical properties such like the muscle in the vagina area, in the vagina environment. And then is the cells vertify on the membrane. And then it integrates the membrane and the chips together to form to the vagina chip platform. So as you can see, this the main structure and the main design of the chips in the middle area. It have the two interlacts the channel, and it can be supported with the liquid such as the nutrition's medium for the cells to grow, and in the middle area of the chips, there at the top right side is the real chips, and in the middle area is the scaffolds. Also is the electrospin membrane. It's the area for the cells grow. For the cells grow. And about the data analysis, I will have a brief induction. First is that I explore I mentioned before, but the middle area of the chips is the scaffolds. Is the material we use is the cathether and the PVA is the one polymer. It's two different polymer material to combine together and using the electrospin membrane. Using electrospin technology to form to the membrane. And this slide I want to show you that I explore the 4 different concentration of the cathether and to see which one is the best for this material to form to the scaffolds. And you can see here, and when the concentration at the 4 is in the SE image, it can be seen the fiber is clearly, and it cannot form to the beast compared to 3 other concentrations. And there is a string stress test for the 4 different concentration membrane. With the concentration of the cathether increase, such as to 2% for 4%, and you can see here the materials can complete elastic deformation at this area. But when the concentration, the cathether concentration reaches to 5, and you can see the elastic deformation is decreased. So why it happens is that it's because the increase with the concentration of the catheter. It means that they have much more polymer materials, and so the more clear weight in material will resulting in greater intermolar force. So it will be reducing the strength of the material. Then is the FTL result. This result is one test for to explore what elements and the material contain with the 2 different materials. Because I use the catheter and the PVA to combine together. I then have the different peak in this. The way as you can see down here is the wave number that in the middle area you can see different peaks in this test. It can be represented. They have different function group which can be represented. They have the catheter or PVA such as I can give you a sample such as is here. In these peaks, it can be represented. There have the hydroxyl and amino group, and it can be like there have the catheter material in these peaks. There is the XRD result, X-ray diffraction. It's also one result for to explore what is the interaction or will happen when the 2 different polymer materials combine together. As we all know, the cathether is the low characteristic polymer, but the PVA is a highly characteristic polymer. So these 2 different polymer integrated together. So it's also as the peak shows, has a different concentration of the catheter with the of the 2 to 5 concentration. And you can easily see the peaks here. And when the concentration of the catheter is higher, and the intensity of the characterization of the peak of the PVA at these peaks, it will increase. And it means that about these 2 materials, catheter and the PVA, they can integrate, and the interaction is can both happen when the combination to these 2 materials together. Then with I mentioned before, I use that electrospin technology to form these 2 materials together to form to the membrane, and then explore another is that using the oriented fiber, electrospin oriented fibers. It's just to change the collected device. And here you can assume this collective device is one rotate is one rotate rollers with one constant speed, and when the when the fiber is free from the needle and it will be collected at the device. So this is the comparison as image with the oriented fibers and the non-oriented fibers. As you can see, this is the oriented fibers is more aligned and have some direction in some contents, and the fiber diameter is much thinner than the non-oriented fibers. It means that in the same, you can assume in the same across section, they have they have much fibers and they can be support much higher mechanical properties. So each the cells we use to culture to survive with the bacterial photosynthesis is a VK2EC7 cell line, and this cell touch on as image cells touch on the membrane, and this is the MTT result for the comparison with the different scaffolds with easily see that in the MTT result of the cells in the Catson PV have the best growth. This is we compared based on the airflow system. We compared as you can see here is our chips and the membrane, and with the airflow we can support with the nutrition and the medium to form with the loop and cultured in the incubator and with this with this loop. And to conclude is that this project is explores the best concentration of the two different polymer material, Catson and PV, to see which is the best to explain and then to verify the mechanical properties can support the vaginal cell screw and to compare with the previous research. All right, thank you. It's my problem.
A question.
I don't think it was very technical setup of the chip.
Yeah.
But I was wondering, are you planning to include in this system also the bacteria?
Yeah, maybe in the further work we will see the bacteria with the whole system and to see when the bacteria infected with the cells, they have some action.
So you want to keep the bacteria separated from the.
Oh, you mean with the device? Yeah. I mean with not separate.
And just curiosity, how can you manage to keep the bacteria contained in the area where you want it? Or the bacteria will start to spread everywhere?
This is a good question. Yeah, because because with the, as you can see here in the middle area is the is the membrane outside, and to support the cells screw, and I think the bacteria maybe cannot control which it can be grow. So maybe we can first we can see the bacteria with the cells on the membrane, but not inside with the chip. Instead with the chip, separate with them.
And how can you control the temperature of this
Temperature of the medium, the environment?
The cell has to be at 27 degrees.
Yeah. This one you inside the incubator.
Everything.
Yeah.
It is one effect. No, okay, not sorry. Yeah, thank you.
Thank you.
Why did you choose Preston with other polymer materials?
Because Catson is one biodegradable and biocompatible material. Firstly, they are not harmful to our real human body. Then is that about Catson, they have much higher mechanical properties and which are suitable for the real vaginal environment. Is a wrinkle maybe in the vaginal environment and the muscle. So it can be support the much higher properties. I mean the mechanistic properties. Thank you.
So I just want to thank all the speakers one more time for some really excellent talks, which were individually excellent, but also I think really well demonstrated the breadth of the research that we've got. We've got physical engineering kind of research, we've got molecular cellular work and population work all going on within the CIRTM. So I think it's been really, really great afternoon. Please don't run away. There are posters on display outside here, and also there were a few questions that we didn't get around to, so please go and talk to the speakers. So thank you again, everybody.